Hi, everybody. This is Miss Peachy from your WCA biology class. And today we are going to be looking at the next unit here, which is Unit 3. So you've completed Unit 2 in Biology A, now you're moving on to Unit 3. And so this lesson is a little bit easier. We're going to be delving into the structure of the cell and the function of the cell, and kind of, how your cells in your body work. So the title of this unit is or this lesson is what goes in must come out. So we're going to be looking at a specific part of your cell. In this unit, we are going to be looking at the cell membrane. So as a reminder for you, on my website, I have a notes guide made up for you. It's just an outline essentially of this unit, where you can fill in the information, the relevant information, as we kind of go through the lesson. So you can have this in front of you. You can use a split screen, or you can print it out and write on it, but then you can kind of follow along with what we're doing in our lesson today. All right. So I'm going to kind of scooch that over to my other screen there. So the lesson kind of begins talking about how there are different types of organisms. There are multicellular organisms, there are single-celled organisms, with so much variety in the different kingdom of living things, with animals and plants and fungi and even bacteria with our prokaryotes. But regardless of the type of organism, one thing that they all have in common is that they all have a cell membrane. And so we're going to be looking at that today, that part of the cell. It's a very important part of the cell membrane. And we have a picture of it up on the screen here. All right, so let's put some notes up here as well. So the cell membrane, it is made up of a double layer of lipids. And lipids is just another name for fats. So one of the essential nutrients that you need in your diet is actually fat. People sometimes try to get rid of fat in their diet because maybe they have too much of it, which is oftentimes a kind of a hallmark of a Western diet, is having too much fat in your diet. But you do need to have fats in your diet. They're essential nutrients. They help build your cells. Without having the fats that are essential in your diets, then you wouldn't have healthy cells. So we can see here in this picture, it's a bilayer, which means a double layer. You've got fats on the bottom and fats on the top, or fats on the inside and fats on the outside, right? And they are joined together, tails touching, heads facing outwards. So the structure of these is such that, well, I should say, first of all, the function here of our cell membrane. So the function is, one of the main things it does is it controls what goes in and out of the cell. Okay, and it also, it's waterproof. So it kind of keeps you protected from the external environment where you're exposed to water and stuff like that, because it keeps the integrity of your cells. You stay together in one piece. Now, as I mentioned, we've got these double layer of lipids here. You'll notice heads and tails here. So our head end is called the hydrophilic end of the lipid. This is facing outwards. And hydrophilic literally translates into water loving. So the basically what it is that one end of your lipid molecule is repelled by water. And since a lot of you is made of water, that end kind of points inwards. And so that's our hydrophobic end. So basically that end of the lipid is repelled by water, so it turns and faces inwards, with the ends of the head ends not being repelled by water, so it faces outwards. Okay, so some other things in the structure of our cell membrane here. We also have proteins that are embedded in our cell membrane. So these proteins have kind of different functions. Some of them are like doorways, like gateways that allow stuff in and out, and some of them are very specialized. So they'll only allow certain things in and out. Some of them act as more like a lock, so that, they have like a specific receptor that's only there for like a certain hormone that has the receptor on the end of the hormone as well, so that they can kind of match each other up. Um, but one such protein is called an aquaporin. This protein allows the movement of water in and out of the cell. Okay, so that's what this one is right here. Kind of showing you the aquaporin protein, which is like a doorway, allowing water to move in and out of the cell. So again, main function, or one of the main functions, other than just keeping you all together and intact, is controlling what goes in and out of the cell. So that's kind of what we're going to be exploring for the rest of today's lesson. How does the cell control what goes in and out of it? So one way that this control happens is through something called diffusion. So diffusion, let me just write that one up here. Diffusion is movement of something from an area of high concentration to low concentration. So in a cell, we are looking at particles or atoms and molecules that are highly concentrated on one side and have a lower concentration on the other side. It's kind of like, you have to think about concentration. What does that mean? Where you have a lot of something to where you have less of something. And I always use the analogy of a slide. So if I have a slide on the playground, right? This is my slide. Beautiful picture. And we're going to go at the top of the slide and slide down, right? Oh, hang on. There we go. At the very top of the slide, you have high elevation, right? So this is high elevation. And this is low elevation. Now, you will always slide from an area of high elevation to an area of low elevation. You're never just going to slide right back up the other way. In order to go from high elevation to low elevation, you're losing energy. You have a high amount of potential energy at the top of the slide and a low amount of potential energy at the bottom of the slide, right? So you're always moving with the elevation gradient, we call it. Well, that's elevation, which we're not really talking about here. We're talking about concentration, but it kind of works the same way. So if we have a cell, and we have our cell membrane here, let's say we have a very high concentration of oxygen outside of the cell. We'll call these oxygen molecules. And a very low concentration inside the cell. So concentration, think about it as the number of molecules in a given space. So if it's a high concentration, you've got more of them crowded into the same space. If you have a low concentration, there's fewer of them. So just like with elevation, these molecules are going to move from an area of high concentration to low concentration in order to come to a state of equilibrium. Once they come to that state of equilibrium, then the diffusion Well, it doesn't exactly stop, but then you're not having like that real diffusion that's occurring, right? You're going to have molecules moving back and forth, but you're not changing the concentration at all. So, diffusion is movement of something. We'll put here molecules and atoms, right? From an area of high concentration to low concentration. So these are typically fairly small things because they go directly through the cell membrane. They're able to pass directly through the cell membrane in diffusion. Okay, so these are small things that can pass directly through the cell membrane. This does not require the addition of energy. We don't need to add energy. This just happens because you already have this state where you have kind of like a differential of energy. You've got high concentration outside, low concentration inside, and so the movement occurs to kind of equalize the energy. So no additional energy is required. So that's why we call this a type of passive transport. So this is one type of passive transport because we don't require any additional energy. Oh, I didn't know I could do that. That's kind of cool. Um, okay. So that's one type of passive transport. So another type of passive transport is going to use those proteins we talked about earlier, the little doorways. So here we looked at these aquaporins, right? And they're like little doorways. So these work for molecules that are maybe a little bit bigger, so they can't kind of diffuse directly through the cell membrane, or in the case of water, they can't pass through the hydrophobic parts of the cell membrane because that repels water, so it needs a doorway to be able to get through. But it still goes from an area of high concentration to low concentration. It just passes through the protein doorway, right? So here we had diffusion. So this is called facilitated diffusion. So facilitated diffusion, same thing as diffusion, still moving from an area of high concentration to low concentration, but it has to travel through one of those protein doorways. Oops. Okay. So again, yeah, I mean, it's still a type of diffusion. It's not something completely different. Now, water is kind of an interesting case. So it behaves a little bit differently. So when we have water traveling across a cell membrane, we have a process that's known as osmosis that's occurring. And maybe you've heard of this one before. So osmosis is kind of a special type of diffusion. This is just applying to water. It's not applying to any other thing. Whereas diffusion is more general, this applies to water. Now, another kind of word that I put here, or phrase, if you will, is semi-permeable membrane. And I didn't really use this one before, but think about what it means. Semi-permeable would mean that it's allows some stuff through, but not other stuff through. So it's not completely permeable in that it can allow anything through, but it's kind of somewhat permeable. And sometimes we use the word selectively permeable instead of semi-permeable, but it's kind of meaning the same thing. Selectively might be a bit more correct, I think, or specific. So with osmosis, we have to understand a couple of different terms. we have to understand what a solvent and a solute are, and a solution. So those are kind of three different terms that sound very much the same. So let's look here at solute. Solute. A solute is something that dissolves in solution, something that is going to be dissolving. A lot of times we think of it being a solid, but it doesn't have to be. It can be a gas. You can even have it being another liquid, but it's the thing that you're actually going to be kind of dissolving. A solvent is the thing that's doing the dissolving. So, like an example here of each one of these. With a solute, like an example would be the powder that you put into Kool-Aid. That's your solute. The solvent is the pitcher of water. You pour the Kool-Aid packet in the pitcher of water, the Kool-Aid packet dissolves into the water, and together, when you mix them up, it makes a solution. So, why this is important is because with osmosis, what we need to look at is the concentrations of our solutes inside and outside the cell and how they compare. So, let's have our cell again. Pretty beautiful. And then if I put in here one of those protein doorways, here, I can make this fatter so that it colors better. Which remember we called these aquaporins before. It's one of those aquaporins. I'm just gonna erase that part. Oops. So normally this would be shut, right? And it's got to open only when stuff can get through. All right, that's all right. So again, if you have, say, we're talking about water, right? Only water here. Say you have okay, so you're talking about water, but you're also talking about a solute. So it's kind of confusing a little bit, okay? But say you have a lower concentration of water molecules on the inside. Okay. So the idea here is, oopsies, you have a lot of your solute, which is the gray, which is in this case salt, on the inside. And you have a lower concentration of that on the outside. So, kind of the best way of looking at this is very salty, and this is less salty. So, we're not necessarily looking at the number of water molecules or the number of salt molecules. We're looking at the concentration of the solution here. So again, this is not in a state of equilibrium, right? So you need to achieve a state of equilibrium. What has to happen for this to happen is we're not going to move the salt molecules. We're going to move water to change the salinity of the solution. So we want to move water out of the cell in this case in order to be able to make the inside. Actually, I take that back. We're going to move water into the cell. So you have to think about it because we're only moving water. So if I move water into the cell, which I got to go through my aquaporin to do. if I move water into the cell, what's going to happen is I'm going to dilute the saltiness inside the cell. I'm going to make it less salty. So if I move water into the cell, I'm going to dilute this. This becomes less salty, okay? And this becomes more salty. So this becomes less here. This becomes more until they're reached a state of equilibrium. Okay, so you're only moving water. So the water either has to dilute. It's basically the water is going to move to the area that's more concentrated, that has a concentrated solution, so it can dilute that. That's essentially what's happening to every one of these examples. Okay, so if I go back to the picture that I have here, okay, this is the type of solutions that we're talking about. So in this solution that I was just mentioning, that's called a hypotonic solution. Hypotonic means that it's more concentrated inside the cell and less outside the cell, right? Our solutions are more concentrated inside and less outside. So water moves in to dilute the inside of the cell. And what happens is the cell expands and will just blow up. So that's not good. In this last scenario here, there is less concentration inside the cell, more outside the cell. So water is going to move out of the cell to try to dilute the more. It's always trying to dilute the more. It makes the cell shrink or shrivel. That's called a hypertonic cell. In the middle example, it's even. So same inside, same outside. So nothing needs to change. Water, the same amount of water flows in as out. So it maintains normal pressure inside of the cell. It's called isotonic. So isotonic means same on the inside as the outside. Hypo means there's more on the inside and less on the outside. So the water will move in and cause the cell to expand. And hyper, we have the more on the outside. So we're actually looking at the solution itself. So hypo means less. So we're looking at the solution. The solution is less outside, so it's hypotonic. The solution on the outside is more, so we call that hypertonic. And then when the solution on the outside is the same, we call that isotonic. Okay, and that's kind of what happens here. Okay, so then this situation is another situation where we call it passive transport, because even with osmosis, it's still moving from an area of high concentration to low concentration. It might not seem like this here, but it really is. We're talking about where you have the concentrated solution, right? So it's highly concentrated inside, so it moves out. Highly concentrated outside, so we're moving towards the area. It actually looks like it's moving from less to more, but okay. So again, we're moving from an area of high amount of water to low amount of water, following those concentration gradients, so it's not taking any additional energy. Okay. Oops. All right, so then our next type of transport is called active transport. And active transport is a type of transport that requires additional energy. So there are some situations where you need to move stuff to an area where there's already a lot of it. And then diffusion isn't going to work. So this requires additional energy. So, for example, if you're going back to that slide example, you slid down the slide from an area of high elevation to low elevation, but then you're going to climb back up the slide to get back up to slide back down again, right? So climbing back up requires energy on your part, adding additional energy. So active transport is kind of the same thing. It's requiring additional energy to be added here, and that's energy in the form of ATP. We'll talk more about ATP coming up here in the next couple of lessons. Okay, so this also then uses these protein channels as well to be able to use the protein channels to move ATP, to be able to give the energy to actually move some of these molecules against the concentration gradient. And then finally, we have a situation where we have very large things that have to move across cell membranes. And so when very large things have to move across cell membranes, they can't fit through those protein doorways. So we have to have a different mechanism to move these in. And that is through endo and exocytosis. So they're pretty much just the reverses of each other. This one here is endocytosis. And endocytosis is where um, larger packets of material or larger molecules inside the cell, as they are actually um, a vesicle forms around them, which is essentially like a mini cell membrane. And a lot of times those are formed like in another structure of the cell called the Golgi apparatus, and they are transported. Oops, I guess I have. Oh yeah, that's our exocytosis. So in exocytosis, the membrane forms around them, and then they move towards the cell membrane. They are incorporated into the cell membrane, and then the outer part of the membrane dissolves, and that material is released outside of the cell. For endocytosis, it's the opposite. So the cell membrane will actually kind of wrap itself around things that need to be transported inside of the cell, forming a vesicle. That vesicle is moved into the cell, and then the little membrane around it is dissolved by another structure in the cell called the lysosome. And the lysosome will dissolve it, it will recycle the materials to be reused by the cell, and then the stuff that's brought into the cell can be used in whatever capacity it needs to be used in. So endocytosis brings stuff into the cell by making a vesicle, and exocytosis transports stuff out of the cell by making a vesicle as well. All right, so that is kind of in a nutshell our lesson for today, Unit 3, Lesson 2. And that's it for this one, ladies and gents, and I'll see you in the next one.